Okay, can you see my screen?
Yes.
Good. Okay, well, thanks very much for giving me the opportunity to talk to you again this year. I must say, when I look back at, last year's, it's amazing how many things have happened internationally during the year and also in my own lab. And probably, even though we're a pretty small group, although I'm a link to another quite larger group that are working on epigenetics and cancer, this has probably been our most productive year. And could I just add that, because I'm sort of have overview of seeing all things like public health policies and responses to these chronic illnesses like ME/CFS and long COVID, how important I think support organisations like MEISS are, to the affected people. It's the stability really that kind of keeps me alive and also keeps, I think, the people who are affected alive from what I get from lots of communications from patients. So, I'm just shouting out a big congratulations for what you do all of the time, for the affected community. Right, so I will just, okay, so we're just not getting, okay, we've got to go to slideshow. We should be right now. Good. So a couple of years ago, I talked about how after 10 years of research, we were able to produce a model, a holistic model for ME/CFS, and we also added long COVID because it had just come on board. It then seemed consistent to what was going on in long COVID. And we used multiple analytical tools for different kinds of molecules in New Zealand patients to be able to derive that. And so this has really driven the research from then on, and really it raised important questions that we wanted to pursue. And so on the model there, I've just put a couple of numbers. And the first question was that the model really threw up and reminded me this was so important, was why is it that 5 to 10% of the population, when they're exposed to a virus or a major stress, they don't react like the rest of the population and resolve it, but they get a chronic effect, a chronic inflammatory immune response and go on to get ME/CFS. So I want to talk a little bit about that today because there's been a lot more happen in the space really just in the last few months. The second thing that our model sort of highlighted was one of the critical core symptoms, of course, is post-exertional malaise, which says when someone affected just does a little too much physically or mentally or has an emotional event in their life which is a little bit more traumatic, then they're very susceptible to getting collapses which can last for days, weeks, or months. So we wanted to actually look at what happens with the onset of post-exertional malaise, and I want to talk about that. And then the other critical sort of change in 2025, rather than looking at different classes of molecules, we focus now on looking at the epigenetic code, and I'll talk about that more, exactly what I mean by that as the talk goes on. But this seemed from that first 10 years of research to give the most stable information across all patients, and we saw this as the best way forward. And for that reason, after 57 years in biochemistry, I switched now a year ago to pathology and molecular medicine, and so I could have a linkage to the group that's got most expertise in epigenetics analysis, and they're working on various forms of cancer. So we kind of standing connected to that group, my small group now, getting their expertise and applying it to the ME/CFS problem. Now, just after we'd published the model in 2022, I was approached by Springer Nature to ask whether I would produce and edit a book on ME/CFS. And this was quite amazing because they generally just in their protocol series publish things on aspects of biology and human biology. So this was, for the disease. And so I was quite excited because it allowed us to put all of the dysfunctional physiology together in one place. In other words, have a holistic view of MECFS. And so what I did was to invite people to write chapters for the book. And for example, I was really excited that we could ask Ros Vellings to write the first chapter on diagnosing how to diagnose an MECFS patient. And we went right through to the current imaging studies that have been done on MECFS patients. And also the last chapter was on the microbiome. So after 3 years of work, this was published at the end of May in 2025. And one of the people, my research fellow of 25 years, Katie Peppercorn, we did it together. And it was very satisfying when this was finished. Now, I don't, the IP for this is in the hands of the publishers. And so they produced this nice hard copy book and it costs $400. So that's okay for university libraries and maybe some scientists and clinicians, but certainly not for patients. But fortunately, an ebook was created and a PDF copy of the ebook could be formed. And so I've been excited that actually patients, not only around New Zealand, but beyond that as well, and some organisations like the Australasian Osteopath Association have contacted me and asked if they could get a copy. And so certainly for patients, I've been able to send out then the PDF at no charge. And so it's been able to be distributed. So if there's anyone listening who would like a copy of that, they just have to email me and I'm happy to send it out. So this really is, this book and the model is given the impetus for what we've moved on to do in 2025. And so what I tried to think about doing is we wanted to have our studies a bit more directly patient-focused or focused on important questions that could lead to an improved quality of life for focus. And that's kind of what we've been doing in 2025. So the first thing I mentioned in the model was, why do 5 to 10% of people go down with the disease? So we've been working on a project then to understand susceptibility in families who have multiple family members affected, either with MECFS and long COVID. And in fact, I'll talk a little bit about that, and the fourth-year medical student is going to come in and extend this now, because we've just started to recruit members of 3 or 4 families to study over the summer to see whether we can develop a family signature, essentially, a molecular signature, which would be a risk signature for those families. Second thing I want to talk about is using our studies on the epigenetic code to look at what happens as post-exertional malaise developing. And we've just had a major paper published in September on this, which we're pretty excited about. It was the fastest publication of any paper that manuscript I've submitted in my whole career. So we were, excited that it was so well received. And then secondly, we've been working on with the epigenetic approach, how closely related MECFS and long COVID is. And we published that paper in July again, and that's been well received. So the data we've got from these studies is really, I think, going to be very helpful to develop a molecular signature which is suitable for development as a diagnostic test. And I'll show you the data and you can make up your mind whether this might be possible or not. One thing we're doing right at the moment, and I'm going to show you one piece of information which is hot off the press, which was the analysis has only been finished this week, and that is that we've always used DNA from a subclass of immune cells, and that's what everybody uses for blood studies around the world. But there's another source of DNA. It's called cell-free DNA and plasma, and I'll explain more what that is. It actually is derived from cells that die and degrade and release DNA, and so it reflects all of the cells around all of the body. And so it has the potential to provide greater insight as to what is happening in other parts of the body with MECFS, rather than just how we've all been focusing on those immune cell, the immune cell DNA and the information from that. So I'm just going to show you one slide from that, as I say, of data we've got this week. I'll show one thing about treatment because, as you probably all know, work is in progress internationally. A lot of it is going on, but it's all really quite confusing at the moment, and we hope the MEISS will clear with that pretty soon. The last thing I've got on my list there is that I was approached by the European Society of Medicine if I'd write an article on a special issue they're having on insights into chronic diseases. And this turned out to be coincidental with an invitation from a GP who's on Waiheke Island, who approached me because he wanted to set up a group of fellow clinicians and allied health professionals, mainly it's in Auckland, people who deal with people affected with chronic conditions. And as a clinician, he's found it very difficult and feels he doesn't have enough information. And so this group has been formed, and he asked me if I'd be part of this group. And so they had their first conference, two-day conference in Auckland. I gave a plenary talk for that, by Zoom, on MECFS and long COVID. But for the article I've just written for the European Society of Medicine, I focused on a wider range of chronic conditions: Ehlers-Danlos syndrome, fibromyalgia, multiple sclerosis, as well as MECFS and long COVID, and included POTS and mast cell activation syndrome, because all of these conditions, chronic conditions, have many overlapping symptoms. And so the theme I wrote about for the European Society of Medicine article was that we should be taking a much more integrated approach to looking at these things and not regarding them in silos, because information that's available from one condition might be actually transferable to others and might suggest more treatment options. And I'm happy to say that actually last night, the article, of course, that I submitted had to go out to international review, but I got, overnight, I got an indication that it's been accepted now for publication. So that's kind of where I've been in 2025 so far. Has been working a bit harder than I'd actually dearly wanted to, but there's a lot of opportunity at the moment to make contribution. So I just want to now talk first about the susceptibility. So our interest started with a young woman, Anna Blair, who did an MSc with me when she was doing a degree in genetic counselling from Dunedin online. She has got an MECFS patient. She was part of my small University of Women's study group that we've done work on. And so her master's project was to do a large survey. It was just when long COVID patients were coming into play in New Zealand, 2022. And she compared long COVID and ME patients with all sorts of different criteria. But one of them was looking at whether they had family members that also had a chronic fatigue illness. And I was quite amazed that we had, I think, about 158 MECFS patients, and almost 50% of them reported having another member of their family, that is the nuclear family plus cousins and aunties and uncles, 50% also having a fatigue illness, not necessarily the same one, MECFS, but a fatigue illness. And with the long COVID ones, 57, she recruited that stage for review, we had 28% had a family member, and most of those were actually members who had also got long COVID. So what she was able to derive from her study was two things: that susceptibility factors, certainly this family medical history, which we call genetic susceptibility, and I'll be talking a little bit more about that in the next couple of slides, but also personal medical history was important, and underlying conditions, comorbidities. So, you know, these things might be that as a child, it took much longer to recover from a childhood illness, or now I've got some conditions and that seems to prime the person that when there's a triggering event, such as a viral infection, such as SARS in terms of the long COVID, or, you know, Epstein-Barr virus for glandular fever, it acts as a kind of a priming, so that when the second trigger event comes, one moves on and develops the disease. So that's where we were at the end of 2022, but there was no information internationally about any links to specific genes. But you'll probably be aware that this year, in August 2025, so far only in a preprint, what's been called the Decode ME study was published out of Edinburgh, and it was the main driver of this was a scientist, Chris Ponting, who had a good friend who came down with ME, and, you know, their promising career was kind of wrecked. And so he was motivated, so really good motivation, to try and do genome-wide, what's called a genome-wide association screen, that is looking at all of the genetic variations across the DNA genome, and we've got about 2 or 3 million between each of us. So just looking at all those variations to see if any of them was linked to ME/CFS. Now, this has been done before, several times, in fact, with about 1,000 patients or less, and no linkage has been found. But the difference in this case, he used 15,000 ME/CFS patients and found genetic linkage to 8 specific genes. And you've probably heard of this already, because there's been enormous publicity about it. Actually, it isn't that profound really yet, but I think, you know, the ME community around the world have been quite excited because they now know that their disease can be linked to specific genes. It actually isn't the first one, and I'll talk about that in the next slide. So there's 8 specific genes. Now, bit of caution one should note with a complex disease like this is that if one has multiple, if one has a disease where there's just a single mutation, and if you've got that change, you get the disease. That's you know, single polymorphism or mutation linked to the disease. But for these diseases, there are individuals in the community that have got these variations in their genetic code, but they don't have the disease. So although these are linked to the disease, not everyone who has the variation gets the disease. So a little bit of caution. So Galina Mandich, the fourth-year medical student, is when she starts next week, is going to analyze the study in great detail, comparing with our own findings. And what we want to do is to look at the DNA that we've got isolated from quite a large number of now ME/CFS and long COVID people, and we will see whether they've actually got these particular genetic variations. However, there was a study actually published first in December of 2022, just after Anna Blair submitted her thesis, which I thought was a major advance, and it was published by a company in Oxford, Precision Life. And what they did was just what the Decode ME/CFS study has done. That is, they looked at the genetic variations, every one. But instead of just looking at them singly, they put in a combinatorial platform where they could look at one and then ask, was this linked to any other variations? And did that form a cluster which could actually link to ME/CFS? And what they found, and this was amazing to me because it came from nowhere to this, was that they found there was 199 variations these single nucleotide, what are called polymorphisms. It's just change in the base at a particular position. And they could group that into 84 families and then 15 clusters. And what I show on here is, and these two are probably the easiest to see, this is a cluster. And you can see in the middle, I've shaded green, the core, which links to all of these other variations. And here's this one here, the core links to all of the variations. So what we've done with this, and so I should say that these 15 clusters are linked to 14 genes. So there was already this a paper which gave 14. These are different genes than have been found with the DECODE study. So that has to be something which we have to resolve, and we're going to try and look at that. So what we've done, and we started this last summer with Bryn Griffiths, who was a summer student, is we got the tools to be able to look at the variation of these green ones, the middle ones, because we thought if we looked at those are the core ones. We have to have those if we're going to have the susceptibility. And so there was 15. So we looked at all, Bryn looked at all 15. And what I'm showing you here is 3 of them, number 1, number 2, and number 15. And you can see with the number 1, when we amplified that region of the DNA up from 5 MECFS patients, 5 long COVID patients, and some controls, we had, in this case with number 1, 3 of our patients had mutation, had that variation. 2 of them didn't. 1 of the long COVID patients did, none of the controls. The second one, we had 2 of the patients we analysed, none of the long COVIDs and none of the controls. When we came to a third one, we were quite excited because he did the MECFS and long COVID first, and then the controls later. We got lots of variations in the patient, but then when we did the controls, we saw it in the controls. And this is over the others that he's done, which I'm not showing here, the controls do show variations. And while this was initially a disappointment for our going on further to actually exploit this data, it's quite unsurprising, because if you have genetic variations which are relatively common, it might be 20% or 30% of the population has this variation. That is what is needed really to have a 5 to 10% susceptibility in a population. So it isn't so surprising. So we've got to sort of accommodate to that when we go forward. So what we're at the stage of wanting to do now then is to recruit family. We've got 1 family recruited already. We're in the process of recruiting 3 or 4 other family members who have the disease and who don't have the disease, to see if we can work out a family signature based around one of these 15 things here. And then we can see whether there are members of the family that have the signature and may be at risk, and others that don't have the signature and therefore would be safe. So that's where we're heading forward, just to see whether it's possible to develop a risk signature for individual families on this data. And just recently, the CEO of the company, Precision Life, has been talking about what they're doing. And he's talked about these 15 communities, and they've gone in further to see what the pathways that are linked to them. And they include sort of subsections of what we know are affected in MECFS, like there's one of the clusters, neural autonomic, another's urea cycle, stress signaling, mitochondrial and metabolic dysfunction, vascular endothelial, immune inflammatory, metabolic stress response, vascular endothelia. So I'm very enthusiastic about this data and where that is going. And so really it is quite a bit ahead, in my view, of the DECODE-ME at the moment, but the DECODE-ME study, with more analysis and more people looking into it in detail, might give us richer information. So I now like to move on to our use of the DNA epigenetic code to look at post-exertional malaise and also the relationship between MECFS and long COVID. So I know all of you won't be thinking about the epigenetic code 24/7 like I am. So let me just explain what it kind of means. So probably you all know we have a genetic code and that we give letters to the 4 unit chemicals that make up our genetic code. 3 billion of them, and these 4 units distributed in various sequences which give us our genetic code, A, G, C, and T. The epigenetic code, and so that's invariant, it doesn't change. We get that from our parents, and it doesn't change during our lifetime. The epigenetic code can change. It's a dynamic code. It can change from all sorts of things that you're exposed to or that you undertake in your life. You can change an epigenetic code. And what this refers to is there are C bases followed by a G in clusters all through the genome, and they act as gene switches to turn on genes or turn off genes or just dim them. In other words, make them a little bit less bright or more bright. And that means the expression of the genes, the information that's being expressed from the genes. So there are regions then spread right through the 3 billion bases, clusters of these, what we call CPGs. They're like lights, banks of lights, really, and they can be all turned on or all turned off, or some of them turned on and some in a particular region. And each region is connected to a particular gene, and so will control how heavily that gene is expressed. Now, the interesting thing is what we found in studying this is that the major type that we see, or major thing that's happening in MECFS, is we're getting a dimming of the lights in these CPG region. In other words, the expression of the genes is mostly going down. And that's consistent with what we know about the metabolism, certainly in the studies we did to work out our model that I presented in the first slide. We know that metabolism is operating at a lower level than in normal people. And that's, we think, is because the body physiology is tending to shut down as a protective mechanism to what it sees as an apparent danger, unfortunately, for the person affected. That means, they're tired and they can't function normally. And this is regarded just like what happens in hibernation in animals when they protect themselves when they go to sleep over this whole period. They don't have any food intake, and so they're relying on a very low level of metabolism to survive before they wake up and resume normal metabolism. The way we've done this, as we've done our other studies, by precision medicine, that we've focused on a few individual patients. But the good thing is now we're starting to do longitudinal studies. That is not just single time points, being able to follow what happens, as you'll see with our post-exertional malaise as we carry on. So for the post-exertional malaise study, we wanted to have a physical exercise stress. And the expert in New Zealand who does this is exercise physiologist Dr. Lynette Hodges at Massey University. So Lynette came down to Dunedin and we set this up in the phys ed school. They have the equipment there. And so what she did during an exercise session, she measured all of the cardiopulmonary parameters, and that's what she's an expert at doing. And the test is a 2 exercise test. So on day 1, they do the test and they have all their measurements taken. And then on day 2, the test is repeated and all of the same measurements are taken. Now, what we did for our molecular work is we took a blood sample before the test, before the exercise test, and then 24 hours later, just before the second test, and 24 hours later after the second test. So we had 3 samples, so we could follow what was happening at the DNA level through from before the test, what had happened after the first exercise, and what had happened after the second exercise. And what I've got up here is just from one patient and one control. We basically were doing this one with just patients, but we did have a couple of controls. We had 2 contrasting controls. One who was a half marathon runner, was obscenely fit, and the other one was what one could unkindly call a couch potato. In other words, was probably a lower than average level of fitness than most healthy people. And so we looked at their parameters, and I'm just going to show you 4 parameters. So I'm showing you one MEC patient and the really fit control, just so you can see the comparison. So the first thing that Lynette measurements was maximum heart rate. And so the MECFS patient on day 1 did pretty well, got to 178, a very fit person, a bit higher. But when the second day came, the patient wasn't able to reduce, get to the same maximum heart rate. And then the next one is maximum work. So what happens is that they're on an exercycle and they have to be able to pedal at a certain rate for it to continue. So when they can't pedal at the same rate, that's regarded as their maximum work rate. And so the MECFS patient had 135 here watts. The second day only got to 120, so was not as good on the second day as the first day. A VO2 peak is how much oxygen you are actually taking in while you're doing the exercise, and this is connected to the use of oxygen for aerobic metabolism. So we're producing energy by utilizing oxygen. So that's quite a good value, and that was still the same on the second day. And then maximum work rate when you switched away from using oxygen to what's called anaerobic threshold, anaerobic metabolism. So let me give you an example of what this means. So if I'm leaving my office and I've got to catch the bus, and it's 5 minutes walk, and I know the bus is coming in 10 minutes, and I stroll down to the bus stop, then I'm using oxygen to do that. I'm using aerobic metabolism. But as so often happens, if somebody stops me as I'm going out the door and wants to ask questions, and I waste most of the 10 minutes I've got, and suddenly I find I've only got a minute or 2 to catch the bus, and I look down and I see the bus coming, then even at my age, I'd try and sprint down to still catch the bus. And then you would be working on anaerobic metabolism. So in real life, sprinters use anaerobic metabolism because they can't get enough energy just from oxygen alone when they're doing 100 meters at 11 seconds or something. So you can see here the switch for the ME patient was at quite a low work rate, 60 of their 135 here that they had. And then in the second day, again, a much lower rate before they switched. When we come to the controls, though, you can see the control day 1 and day 2, maximum work rate exactly the same, really high. I mean, this person was working very hard and was relatively fit and could keep going for a long time. VO2 improved a little bit as well, just like the heart rate, and maximum work rate before she switched to anaerobic metabolism was improved as well. So that's not actually surprising, Lynette finds with healthy people, is that they kind of get used to the exercise, and then when they do it on the second day, they can do a little better, and that's what we found. But even the control who was unfit, and you got initial figures rather more like this, not necessarily as low as this for the anaerobic. she had exactly the same figures for the second day. So we're seeing, an inability to maintain, the exercise protocol if it's done 24 hours later. So we wanted to look to see, okay, what did that mean at the molecular level? And so for this, I just want to talk to you about a picture that I'm going to show 2 or 3 of these kind of diagrams. So what here, we see this goes from 0 to 1. If it's at 1, it means there's a bank of light switch linked to a gene. There would be a new gene number here, which have been turned out. So this is actually gene expression off. We call that hypermethylation. At the other end, when it's 0, it means there's no of these chemical tags or off light switches, and so therefore the expression is as high as it can be. So that's, if you like, the lights on, that's the lights off, or gene expression on, gene expression off. And so this is what you'll see this in the next slide. So here's this longitudinal thing that we're seeing with all of our patients, and this bank of lights linked to a particular gene, and what we see is before it's about halfway between on and off. At 24 hours in the sample, you can see that it's become nearer to off. In other words, there's been a change from here to here. And then from 48, there's been another change. And so it's tending to dim down the lights, turn off the gene expression switches in response to the exercise. This was the most predominant pattern that we saw. But let me now just show you there are 4 patterns in 2 sets of them, and this is the mirror image. And so here's this diagram I just showed you here, going from 0 to 1. We see it going, the gene expression of this gene becoming lower as we go on from the 24 and the 48 hours. So that was the most predominant one. We also saw a pattern where there was a change at 24 hours and then no more at 48. And we saw a pattern where there was a change at 24 and it actually reversed at 48. And we saw a pattern that was nothing happened at 24, but then it changed at 48. And so when we look at the other set, you can see it's going in the opposite direction. So here, the gene switch is being turned on more. So the light is becoming brighter at 24 and 48. So it's the mirror image of that. And this one goes at 24 and then no more. And this one, you can see it's almost fully on, goes to fully on, and then there's a reversal. And this one, nothing at 24 and 48. So we got a rich set of data. In fact, what we found, and this was amazing to me, there were 209 of these kinds of sites linked to genes. Only 4 of them were in the controls. In other words, there were 205 of these, which were unique, what we call differentially methylated fragments for the ME/CFS patients. The control group had their own ones, their own unique ones, and they were about adjusting muscle function and things like that. So they were responding almost positively to the exercise and adjusting physiologically. But the ME/CFS patients, by these changes and these unique things, were adjusting to really a danger response. And I like to see some positive out of this. The positive thing is that when we looked at physiologically what all of these things, these 205 were linked at, they were linked at the immune dysregulation and inflammation. And that's just the thing that happens when you're exposed to your first trigger that leads to the disease. So what this is saying is that the body has this physiological response to stress, not only from the first trigger when the disease comes on, but any time when there are perceived stresses during the course of the ME. And so when there's in this case, exercise stress, they came down with that. So this is the article we published in September. Okay, so what this has told us is that even though we're looking at only a small number of patients, all patients have the same types of changes. And it seems possible that we could have a molecular signature from about maybe 100 of these regions. I've just told you we saw 205 and changing in response to exercise. So we could take maybe 100 of these regions, and that has the potential to give a diagnostic test of high specificity and high selectivity. What that means is specificity. So you would diagnose 95% of the people that have actually got the disease, and selectivity means you wouldn't get false negatives. Not only, and I'm going to show you this data now, we could have a test that would recognize both MECF and long COVID together. And why is that important? Because now, of course, no one's testing for COVID, so that when someone's get a flu-like infection and it goes on to have an ongoing disease, there's no way of knowing whether that's from COVID or whether it's from Epstein-Barr or anything else. So the long COVID people of the future will be just like ME/CFS. They'll be desperately trying to get a diagnosis for their disease. So having one that can recognize them both together would be good. But it's also good to have a test that could differentiate them, because it may be we'll find some treatment options better for long COVID that aren't so effective for ME/CFS. And so it would be good for patients to be able to know if, in fact, they did have a COVID infection which gave their disease. Now, the critical thing to me about a diagnostic test, there are lots of claims of biomarkers in the literature, and even just recently, a couple of weeks ago, a new claim for potential diagnostic test, which may be complementary to ours and could be, but the critical thing, this has to be accessible to patients and their clinicians in all community pathology labs. And what we're talking about here has the potential to be adapted with a relatively new kind of sequencing or nanopore sequencing. So the data can be on a memory stick, a USB stick, and you can actually analyze that on a laptop computer. So it would be possible eventually to have every community lab with the ability to actually do these tests. Right. So I want to say a little bit now about long COVID and how is ME/CFS and long COVID related. So just a few simple facts. Just to remind you that long COVID is the popular name given to the post-viral condition that arises from just the single trigger virus, SARS-CoV-2. ME/CFS, of course, comes from multiple triggers. 80% probably viral, but 20% from other major stresses in one's life. So that's a difference between classic long COVID patients that we know have been connected with the virus and ME/CFS. However, long COVID is actually heterogeneous. And so the names given to anyone whose disease or whose symptoms just don't improve for up to 3 months. And we know it's heterogeneous because certainly with the earlier variants of the SARS virus, there was quite damaging effects to the lungs of some people, particularly those that were hospitalized. So in 2023, fortunately, a group in Italy did a multinational group. In fact, they took 2,000 long COVID patients, European patients, and from that early group, tried to distill out what the heterogeneity was. In fact, they found about half of them, what I'm calling, had an ME/CFS-like syndrome. 23%, about a quarter, had a respiratory syndrome coming from lung damage which was what those variants were doing. About a fifth had a pain syndrome, and I see that just like fibromyalgia. And then there was a specific 10% with a neurosensory syndrome, and they related that to the loss of taste and smell, which you may remember was something which people were saying was specific to the SARS virus. But the critical point here is that when long COVID patients, when the World Health Organization was evolving the clinical case definition, they were asked to report all their symptoms. They reported over 200 symptoms, just like MEC CFS patients have, and 95% of them overlapped. And certainly in Anna Blair's studies, you found that all of the symptoms that they were reporting were overlapping. So that suggests the two conditions are very similar. But we wanted to get evidence of that from the epigenetic code. And so what you're seeing here is these same kinds of diagrams I've shown before, but in this case, these are single time point studies, and they involve healthy controls, long COVID, and MECFS. And I should have said before, both the other study, I think I had Cheyenne's picture in the other study, and this is my Indian PhD student that's been with me for a year, and we've now published 3 papers together. He's an amazing student, worries about every result and disturbs me very frequently to talk about it, but it's amazing to have students like that. So he analysed these, and these are examples of the types of data we got when we were comparing MECFS and long COVID. So the gene names are here. So this is a bank of these gene switches which are linked to this turning on this gene or turning it off. And we can see down here, we have the healthy controls. So there were 5 patients, and we can see a tight group of them, and their switches are at the same position. And then with long COVID, we can see quite a big change heading towards turning off the light switches. And an MECF is also heading off. Now, this kind of pattern was very common, but actually in many cases, the long COVID was a greater change. And we think that might be because it was the stage of the disease. All our long COVID patients had only had their condition for 1 year, whereas the average for the age, these are all age sex linked, healthy controls, long COVID MECFS. The MECFS, their average length of illness was 12 years. So we think the juvenile form, if you like, of long COVID, there might be a greater response. So that was the most common one. About a third of frequency, we had them going in the opposite direction. So here, again, middle kind of dimness, and now the gene switch is turned on in both of the conditions. Now, what this says is we have signals which are happening in long COVID and MECFS, and so we should be able to develop a molecular signature for an early diagnostic test from these kinds of signals. We also had some rare signals where only one of the disease sets changed. Here's long COVID changing, going towards turning off. The MECFS didn't have any effect on this gene at all. And we even had 1 or 2 where long COVID would go upwards and the MECFS would come in the opposite direction. So these are ones where we knew there were some differences between long COVID and MECFS. So as I think I said earlier, we published this paper in July of this year. Now, so because we'd shown that there was some of these sets of genome CPGs, the light switches, where there was a difference between the two, we actually searched for those to see how many there were. And so we examined 77,000 fragments with these banks of switches, fragments with tags. And all of the fragments were present that we detected in the MEISS patients, the long COVIDs and the healthy control. So we only took the fragments which we could detect in all of the 15 patients. And we said, are there significant differences with 95% confidence between MEISS and long COVID? How many of these had significant differences? Actually, we got 4% had significant differences, and that's actually in a number 2,636, I think, of the 77,000. Now, we can show this graphically in what's called a principal component analysis, where we're looking at where the variation in the data fits in 2 dimensions. And so here we see our healthy controls up here, and you can see they're all clustered together, and they're well separated from the ME, and they're well separated from the long COVID. Now, that's not surprising because we set this up to select that there had to be a significant difference between healthy control and ME, and there had to be significant control between the healthy and long COVID, and there had to be a significant difference between the long COVID and ME. So you can see the ME group here are quite clustered again, and the long COVIDs are clustered. So we're pretty excited about this clustering in terms of potential for an early diagnostic test, because it says all of the changes in the long COVID are found in all of the 5 patients, or pretty well all of them in all of them, because they wouldn't form this tight cluster. With MECFS, we've got one patient here who stands a bit on the sideline, and she's a young woman who, on all of the tests we've done with her, she's a little bit, doesn't actually get in the center of the group. She just stands there, but she's still within the cluster. And you can see one healthy control here is a little bit out, and so I was wondering, well, how come that healthy control is kind of moving away from the rest of the cluster? Well, it turns out the age of these patients, these were all about the same age, and we had one elderly patient as a long COVID, and so we had to have an elderly patient as age-sex match control. It turns out that it's known the epigenetic code does change gradually with age, and so we think the reason we're seeing that just drifting away is because we're actually predicting, we could predict that's an elderly patient, and she was an elderly patient. Now, I just want to move now to the last couple of things, and this is the new data we've just analysed this week. Again, this is Cheyenne's current work, and he's going to talk about this giving an invited talk for emerging researchers at the International ME and Long COVID Conference in Australia in 2 weeks' time, and I'm also giving a talk there. So what we wanted to do was to look at this new source of DNA, the plasma cell-free DNA, which is present as small fragments of DNA, which can come from cells around the body, which have degraded and ends up in the blood. And so what we've done here is compared that DNA with the DNA we've isolated traditionally from a subgroup of the immune cells to see if it gives what signals it gives. And so I'm just summarizing this here. So what we see up here is the healthy controls, the same healthy controls that we isolate both types of DNA from. They formed here with the cell-free DNA, and they formed down here from the immune cell DNA. And so what it's saying is that it's confirming that the DNA here represents different cells than here, because we know that different cells in the body express genes in different ways from other cells. So we are seeing, this is reflecting different cells than our traditional ones are. But then when we look at the ME patients, we see that generally they've clustered. So here's our ME patients here. And again, we've got one of the ME, in this case, a bit of a outlier. And the long COVID patients sorry, the ME patients, this is not long COVID, this is all ME, the ME patients from the cell-free DNA, they're close, but they're just subtly separated. So this is saying we might not get much more data from the cell-free DNA than the traditional thing because they're close together. And what we've found with this kind of clustering of the healthy controls, which you see is more less clustered than the others, this may reflect that each of us will have a certain rate of degrading our cells and dispensing the DNA from that in small fragments into the plasma, but there might be quite a bit of variation from each of us, which is not seen when you're looking at DNA from a particular cellular type. So at the moment, we think probably this isn't going to be a superior kind of DNA. It might actually be provide some complementary information, and I've only made that kind of decision in the last 24 hours. Now, to finish off, I'd just like to say one thing about treatments. This study was published by a patient foundation, the Open Medicine Foundation in the United States, in July of this year, and it surveyed 4,000 patients, MECFS and long COVID, who had been exposed to 150 different treatments, mainly drugs and supplements, some other modalities like pacing's on the list here, and vagus nerve stimulation is on the list. So what they have done here, and it's a complicated slide, but I just want to focus on this over here once I've explained what this is. So here are the top 20 odd of the 150 in terms of effectiveness. So the best drugs or procedures that gave the best effect. Now, I want you to look at is this section over here, and you'll see this starts on the left with bright orange, and then pale orange, and then sort of yellowish and grayish and blues. The orange are the proportion of patients across here that got quite a lot of benefit from the particular treatment. The pale oranges, they've got some benefit. The stuff in the middle really is no change at all. And over here, those that actually were made worse, their symptoms and their health status was worse from taking the treatment. So, I'm an optimist mainly, but it's kind of disappointing when you see that the best treatments available at the moment, things, melatonin's on this list and pacing's on this, and low-dose naltrexone's on this list somewhere. Here we are here. So these are certainly the top, but the proportion of patients that are getting significant benefit, you can see is pretty modest. So we've still got a long way to go to the way we manage treatments. And I think this is, of course, is because each of the people affected are their own clinical phenotype. And unfortunately, what has to happen is each individual has to try things systematically and find what works best for them. And as we get new treatments, that'll be the case. And some of them will probably work for some people and won't work for others. So this pattern will repeat, but hopefully if there's 4 or 5 things that you could do, we might be able to get these orange bars coming up to a much greater amount. And to finish off, I just want you to look at this thing down the bottom here, which is in contrastly very blue. And this is graded exercise therapy. So you'll know for many years in the United Kingdom, the NICE guidelines said that graded exercise therapy was the preferred treatment for MECFS. In New Zealand, there's still pockets of clinicians who are promoting this. It was on the general practitioners' guidelines as the still suggested treatment, and I think may now be taken down. Doctors that insurance companies use, say that graded exercise therapy can improve the person's position, and the insurance companies say unless you do this psychological cognitive behavioral therapy and graded exercise therapy, they won't continue insurance. And yet the evidence says, if we look at the blues, 75% of the patients, said that they are their condition was considerably worsened by graded exercise therapy. And you might be able to see a thin sliver here of orange, which there's a few people who got significant benefit. Now, I'm not against exercise, and I think exercise, when the individual is controlled, I mean, within the guidelines of pacing, I think is a good thing if they can manage it. But certainly the systematic graded exercise therapy programs have been very damaging, clearly, to affected people. And I just want to acknowledge, still I have close contact still with Ros Vellings, and we discussed lots of things together, and so I'm grateful for that. She's been a real inspiration for my work. And then the two people underneath that are the experts in the epigenetic analysis, whose group I'm an honorary member of now, and really grateful for ongoing financial support from MEISS. Just like MEISS, it's very hard to get research funding, and each patient I analyze costs over $1,000 now. So it's, we kind of are constantly living off the smell of an oily rag and just having to get more money as we progress the research. But we've been able to do it. I'm very grateful to certainly private donations from individuals and families, that just when we think we're at our wit's end, someone will give us $10,000, and so we can do another study and study another 10 patients. So, and of course, I'm really grateful to all of the MECFS and long COVID study participants who participate. And the great thing about doing these studies with small numbers of patients is you can build up relationships with the people and be communicating with them. And I find that is just such a privileged position to be in. And that's really is so uplifting for me when we get good results, that we can at least report them to individual people who have been part of the studies, and they're excited to get that. So I will finish there, and thanks very much for your patience and once again listening to me.
Well, thank you very much, Warren. And now it's time for any questions for Warren.